I'm so happy to introduce Dr. Alan Moses. Alan has expertise in multiple areas of diabetes. He's been a frontline clinician and also served as the chief medical officer of the Joslin Diabetes Clinic. He's worked in both basic and clinical research and put this experience into action at Novo Nordisk, where he served several roles, lastly, as the global chief medical officer. During his time at Novo Nordisk, Alan played a really key role in the development and regulatory approval of next-generation insulins. That includes insulin degludec, Tresiba, faster Aspart, and the development of long-acting GLP-1 receptor agonists, liraglutide and semaglutide, that demonstrated their excellent glucose control and this added benefit of cardiovascular protection. To me, the most gratifying contribution that he's made to the world of diabetes is with his wife, Liz, also so highly admired in our field as a parent of a child with diabetes. He's always understood so well what that role is like, and with that, what it's like to be a patient. He's served as a vital member of the board of directors of the diaTribe Foundation, which greatly benefits from his unmatched expertise. And I will now turn it over to Dr. Alan Moses.
100 years ago today, Dr. Frederick Banting, a general surgeon, and Charles Best, his medical student assistant, were sweating in a hot laboratory at the University of Toronto, trying to remove the pancreas from a dog who would be studied with a pancreatic extract that they had been working on to reverse diabetes. We've come a long way over these 100 years, from rescuing individuals with type 1 diabetes from certain death through the first use of insulin, to discussing how the new science of 2021 raises the possibility of preventing, delaying, and or even curing type 1 diabetes. Drugs that may delay the onset of clinical diabetes by modulating the dysfunctional immune system of people destined to develop T1D have become a hot topic of discussion, both at scientific meetings and in the lay press. At the same time, we are seeing bright light shining on replacing insulin therapy with engineered cells that replicate what normal beta cells do in a pancreas not affected by diabetes. Stem cell biology now has brought us to the time of testing different cell lines in various configurations to provide clinically relevant insulin secretion. And this is all on the background of major advances in technology for measuring blood glucose in the human body continuously and coupling those measurements through sophisticated mathematical algorithms to pumps that administer insulin, and in some cases glucagon, when needed to achieve previously unobtainable blood glucose control, or simply put, more glucose levels in the range of individuals without diabetes, now referred to as time in range. So, will the new biology win out over the new technology? What hurdles remain that we need to overcome? How does hope balance with clinical reality as we test these treatments and apply them to ever more individuals with T1D? How can we identify individuals at risk for T1D before they become ill? How do we recruit individuals to participate in clinical trials in order to test these new potential treatments? How quickly can we intervene with adjunctive therapies beyond insulin at the diagnosis of T1D? Is cure of type 1 diabetes possible? And if so, how does one define it? These are some of the issues that we'll explore over the next 90 minutes. To do this, we have gathered 4 individuals with diverse and highly relevant backgrounds. Each of our panelists is deeply invested in improving outcomes for individuals with type 1 diabetes. 3 of our panelists are actively engaged in research activities at companies striving to develop new treatments for T1D. In some cases, they may not be able to speak specifically about the drug in development because of restrictions based on regulatory review. Our final panelist is a world expert in diabetes treatment and diabetes technology, and an individual whom I will rely on to help translate some of these exciting treatment possibilities into practice. I'll introduce each of the speakers briefly and ask them to provide some background on what their companies are doing to alter the course of type 1 diabetes. Then we'll have an open discussion about some of the questions I've just raised. Audience members also will have an opportunity to ask questions toward the last part of our time together. Please ask your questions in the chat function, and I'll have an opportunity to address the entire panel or specific panelists based on your questions. We'll do our best to get to as many of your questions as possible. Our first panelist is Dr. Henry Anhalt. Henry currently is Executive Director of Medical Affairs at Provention Bio, a relatively new company on the forefront of developing a therapy to delay the onset of type 1 diabetes. I've known Henry for many years. He's the consummate clinician, sensitive to the needs of his patients when providing clinical care, and dedicated to improving outcomes for all with T1D. He also has vast experience in pharma on both the medication side and the device side related to both type 1 and type 2 diabetes. Henry, the microphone is yours. Why don't you provide our audience with some insights into what we now accept as the stages of type 1 diabetes and the importance of identifying these stages correctly and how Provention Bio is positioned to make a difference in T1D. Henry?
Thank you, Alan, for your very kind words, and thank you to diaTribe for giving me the privilege to be able to address this audience today. Many years ago, when I went into pediatric endocrinology and realized that diabetes was perhaps the single most important disease that pediatric endocrinologists could take care of, because at the end of the day, I couldn't see any other disease that required so much effort on the patient and family's part in order to manage and live with that disease. No other disease brings that same burden. So I'm delighted to be at Provention Bio and to share with you some insights that I think set the groundwork for the conversation that we're going to have today, and that is about the reclassification of the autoimmune nature of type 1 diabetes. Up until 2015, the diagnosis of type 1 diabetes was only made when hyperglycemia, and unfortunately, and up to about 40% or more new diagnoses, are accompanied by diabetic ketoacidosis. This occurred on a background of typical symptoms of increased thirst, frequent urination, and weight loss, despite increased caloric intake. Need for insulin was immediately recognized, and a life of insulin dependence began. Fortunately, in 2015, the field opened up quite dramatically when the JDRF, the Endocrine Society, and the American Diabetes Association established a new classification system that defined type 1 diabetes in 3 stages, with stage 3 being the clinical scenario I just described. Stage 1 is defined as having 2 or more diabetes-related autoantibodies representing damage to the beta cell with normal blood sugars and no symptoms. Stage 2 is when you have 2 or more autoantibodies and mildly abnormal blood sugars, also called dysglycemia, that people wouldn't even know they have. And this would be a situation where the fasting blood sugar would be 100 to less than or equal to 125, a postprandial or after meal blood sugar of 140 or less than or equal to 199, with a hemoglobin A1C of about 5.7 to 6.4, still with no symptoms. And stage 3, defined as having 2 or more autoantibodies, symptoms, and blood sugars high enough to meet criteria for clinical type 1 diabetes and insulin dependence. Here, it's a fasting blood sugar greater than or equal to 126, 2-hour greater than 200, an A1C of greater than 6.5, or a random glucose at any time of greater than 200. The key takeaway here is that even in the presence of normal blood sugars, once you have 2 or more autoantibodies, you have type 1 diabetes. The implications of this are really important and vast, and we'll have a chance to talk a little bit more about that in just a moment. This shift in classification fundamentally changed the understanding that stage 3 is the result of the disease of the immune system that could have been detected earlier by screening for autoantibodies to insulin, all evidence of decreased functional beta cell mass. And important also to note that those in stage 1 have nearly 100% lifetime risk of developing stage 3 in the scenario that I originally started my remarks with. Identification of those with stage 1 or stage 2 is dependent on screening, and that's highlighting one of the most critical reasons to screen for autoantibodies. Let's drill down a little bit on the differences in population risk. In the general population, the risk of developing type 1 diabetes by age 20 is about 0.3%, fairly rare. Having a first-degree relative with type 1 diabetes increases the risk approximately 15-fold, resulting in an approximately 5% risk. The lifetime risk of developing type 1 diabetes among the general population is estimated at 1%, whereas the risk among individuals with first-degree relative with T1D is 10 to 25%, depending on who in the family has type 1 diabetes, both parents, a mother, a father, or other members of the family. Despite the increased risk in relatives of individuals, about 93 to 95% of patients with type 1 diabetes do not have an affected relative, and that's what we will hear. No one in the family did. Therefore, limiting screening to relatives alone will miss that very, very important and large group. In the absence of commercially available treatments, guidance has always been to obtain screening for enrollment in research trials alone. Let's turn for a second to explore what other reasons there may be value in screening. How about DKA that I mentioned at the very beginning? DKA, which occurs too frequently in people newly diagnosed with type 1 diabetes, require immediate hospitalization as it can be fatal. Furthermore, patients with DKA diagnosis have been shown to have significantly worse glycemic control over time. Knowing the stage is an opportunity to educate on signs and symptoms and provide anticipatory guidance to a family before a life of insulin dependence occurs. In addition, identifying those at stage 2 provides opportunities to enroll in clinical trials and potentially benefit from immunomodulatory and other interventions when they are commercially available. With the goal of modifying the immune trajectory and course of the disease, yes, we are living in a very, very exciting time for treatments in type 1 diabetes. A bit about Provention Bio. Provention Bio has filed a biologic license application for teplizumab, which is an investigational anti-CD3 monoclonal antibody being developed for the delay of clinical type 1 diabetes, stage 3, in at-risk individuals. The submission included results of the pivotal TrialNet TN10 study, a phase 2 randomized placebo-controlled trial that a single 14-day course of teplizumab delayed insulin-dependent clinical stage disease by a median of at least 2 years in pre-symptomatic patients with stage 2 T1D compared to placebo. These results were published in the New England Journal of Medicine in 2019. We are also evaluating teplizumab in patients with newly diagnosed insulin-dependent T1D in the phase 3 PROTECT study. Our approach is to seek ways to address autoimmune disease before symptoms and clinical consequences present, bringing to market medicines that target upstream autoimmune pathways and even viral triggers to fundamentally alter the autoimmune disease process. A revolutionary thought in pharma today when we are managing and treating after the fact. Additionally, our commitment to T1D is reflected in our pipeline. In addition to teplizumab, we are in clinical development of PRV-101, an investigational polyvalent Coxsackie-B vaccine, which we are developing as a potential vaccine for acute Coxsackie-B infection and for the potential prevention of up to 50% of T1D cases. It's also been identified as a likely environmental trigger of the onset of celiac disease as well. Thank you.
Thank you so much, Henry. I think you really got us off to a great start in terms of setting the standard of what type 1 diabetes really is, how it's changed over the years, and some of the excitement of the development of new drugs to delay the onset and potentially mitigate the consequences of the disease early in its course. I'm now going to turn to our next panelist, who's Dr. Georgea Pasedis, who leads global medical affairs for Dompé Pharmaceuticals, where she is Senior Vice President, Global Head of Medical Affairs. Georgea is a doctor of pharmacy and a certified diabetes care and education specialist. However, unlike Provention, Dompé is not a new company, having begun operation in Italy over 80 years ago. But it is new to diabetes and relatively new to the US market, where Georgea has played an active role in developing its profile. Dompé also is pursuing a therapy that impacts the immune system and is meant to alter the course of early type 1 diabetes. Georgea, please give us a better sense of your background and provide us some insight into what Dompé is doing in diabetes and how it's working to expand its activities in the US. Thank you.
Thank you so much, Alan. I am so honored and excited to have the opportunity to be on the panel and echo Henry's sentiments, and thank you to Kelly Close and the diaTribe team, and really feel honored to be among such a prestigious group of individuals. To jump right in my role as Senior Vice President of Global Medical and Clinical Affairs here at Dompé, I have the pleasure of overseeing all our therapeutic areas and countries, with our current focus and commitment being type 1 diabetes research, really looking to find new novel approaches to treating patients. Like you said, Alan, Dompé is not a new company. Dompé is a 130-year legacy company, privately owned and started in Milan, Italy, which is where I am tonight, with offices in Boston and San Francisco, as well as recent offices opening up in China, Beijing and Shanghai. Dompé really began as a large chain of pharmacies across Italy with a rich history of innovation. Our portfolio really is broad, ranging from products within the primary care space, with our advancements in biotech, specifically in ophthalmology, which is where we're commercialized today, and a vast pipeline in oncology, COVID-19 pneumonia, as well as our current commitment to type 1 diabetes. And maybe to just give you kind of the high level overview or an idea of where we started to where we are today, our most recent innovation at Dompé was our clinical trial program in ophthalmics. And we brought a product to market called Oxervate, which was for a rare disease called neurotrophic keratitis, which causes corneal ulcerations. And it really is a vision-threatening disease. And really, Oxervate was our first opportunity to bring a novel recombinant human nerve growth factor to market, really, which is just a perfect example of Dompé's dedication to rare diseases, bringing novel innovations and therapies to patients with high unmet medical need, which has really brought us to where we are in type 1 diabetes today. And similarly, in this space, Dompé is seeking to have a really course modifying medication through a novel new mechanisms of action. We actively are enrolling our phase 3 study in new onset type 1 diabetes with our novel investigational molecule latorixin, which is a CXCR1, CXCR2 inhibitor, which ultimately has its biological activity on the inhibition of interleukin 8 at the receptor level. And as we all know, interleukin 8 is a pro-inflammatory mediator involved in the innate immune response. As Alan, you alluded to, our ultimate goal is to focus on really having that immune-mediated loss of functional beta cell mass being targeted, focusing on how we can make an impact in this subset of patients. Based on results that we presented at ADA and EASD last year, our hypothesis is that the novel mechanism of action of oral latorixin aims to alter the course of type 1 diabetes by slowing the disease progression through the preservation of beta cell function in persons really recently diagnosed with type 1 diabetes who have that low residual beta cell function upon enrollment. And so as we're continuing our commitment and expansion of research in new onset type 1 diabetes and bringing innovative therapies to market for patients earlier in the disease continuum, we really are going to continue our collaborations with hundreds of research centers. Our phase 2 and our phase 3 trial are currently in research centers across Europe, the US, Israel, And we really are hyper-focused on being able to bring new innovative therapies for patients with type 1 diabetes.
Great. Thank you so much, Georgia. I mean, first of all, it is just wonderful to see new companies entering the diabetes space. Those of us who have worked in this area for so many years and have seen, unfortunately, too many companies disappear from the diabetes space are delighted to have you join us and wish you the greatest of success in your endeavors. So we've now heard about two companies and two different agents, both of which are targeting the immune abnormalities of early type 1 diabetes, either before the clinical onset or, as you've just heard from both Henry and Georgia, at the time of clinical onset. We're now going to move a little further along that spectrum of T1D because our third panelist helped to develop the procedure for taking human stem cells from an undifferentiated state to the stage of producing insulin and releasing it in a physiologically relevant manner, that is, in response to glucose. Dr. Felicia Pagliuca is a PhD and Vice President and Disease Area Executive for type 1 diabetes at Vertex Pharmaceuticals. She's also a member of the Vertex Cell and Genetic Therapies Leadership Team, where she leads the cross-functional type 1 diabetes disease strategy team. Dr. Pagliuca moved to Vertex with the acquisition of Sema Pharmaceuticals, which she co-founded based on her discoveries in the laboratory of Dr. Doug Melton. Vertex, which made its name in developing therapies to reverse the consequences of the underlying genetic abnormalities of cystic fibrosis, is also new to the diabetes field, but is positioned to be a major factor in beta cell replacement. Felicia, can you give us some insights into how you began your work in cell differentiation and islet cells specifically, and the goals that Vertex has for treating type 1 diabetes with islets developed from stem cells?
Well, thank you so much, Alan. It's really such a pleasure and such an honor to be here speaking with all of you tonight. And as Alan pointed out, I am a scientist and an entrepreneur and have spent my entire career studying cells, studying the basic building blocks of the human body. And in type 1 diabetes, we have the unusual situation where one specific cell type, this insulin-producing beta cell, is lost, is destroyed by the processes that Georgia and Henry spoke about moments ago. And so our vision is to figure out ways to recreate those cells and replace them in people living with type 1 diabetes. So that work began, as you alluded, in academia at Harvard in the lab of Dr. Doug Melton, as well as many other folks in the field who have been studying this approach of turning stem cells into insulin-producing cells. And we started Sema entirely for the reason of bringing that science out of the academic laboratory and translating it into potential new therapies for type 1 diabetes. So because of the work that we had done in this space, we became part of the Vertex family about 2 years ago. And it's been a really exciting transition for us because, as you noted, Vertex has a long history of focusing on diseases where the underlying causal biology is well understood, like cystic fibrosis and like type 1 diabetes. And so I think when you think about where we are today, there's a lot of excitement about cell and gene therapy in the biotech field. There's a lot of buzzwords out there. But what's remarkable is that type 1 diabetes is one of the few diseases where folks are working on cell therapies where there is clinical proof of concept already that it can work, that a cell therapy can work. And by that, I am referring to the clinical experience of organ donor-derived islets and islet transplant that clinicians and patients have pioneered over the last 2 decades. And so we've learned that if we get it right, if we get high-quality islet cells, if we protect them from immune destruction correctly, that we can render people living with type 1 diabetes insulin independent for many years. We've been very fortunate at Sema and at Vertex to have many friends in the community living with type 1 diabetes talk to us about their experiences, including people who've gone through an islet transplant using these organ donor-derived islets in the early days. And I'll never forget one woman, a fellow mom, tell me a story about taking her young kids to a shopping mall and experiencing a severe hypoglycemic event that left her loss of consciousness and her kids alone. And she ultimately underwent an islet transplant procedure and speaks about it as a whole new life. It was successful for her. Of course, it isn't in all cases, and that's part of the work we have to do. But for her, she went from that kind of concern and burden, as Henry so nicely pointed out, to a life where she can check her blood glucose for fun, which is, I think, for us, something we hope to deliver for everyone living with type 1 diabetes one day. So when you think about where we are today with the science, we're now able to, for the first time, produce from stem cells fully differentiated insulin-producing islets, including those all-important beta cells. And that has given us an opportunity now to bring that type of therapy into the clinic for the first time. Vertex has an open clinical trial for its first cell therapy using these stem cell-derived islets, so we're very, very excited about that. I think, as many of us know, clinical trials are really the bridge between breakthrough science and eventually developing approved medicine. So very, very exciting to be at a stage now with bringing that therapy as a new option for certain people living with type 1 diabetes, and in this case, severe hypoglycemia. Of course, this is only the first step in a journey to developing therapies that could be a potential new option for many, many people living with type 1 diabetes. And we know that dealing with the immune system component is the other half of this puzzle. One, figuring out how to make the cells, which we're really excited to be at that point. The other is figuring out how to modulate the immune system. And in our pipeline at Vertex, we have work ongoing with what are called encapsulation strategies, where we can protect the cells by putting them inside a device that protects from the immune system. And we're in late stages of preclinical development with that program. Very, very exciting work. We have research ongoing, as you alluded, in modifying the cells themselves to mitigate this immune destruction. And I think it's really, as a scientist working in this space, never been more exciting in terms of what we can do today that really, when I started my career, were considered a bit of a dream at the time. So very exciting, and really, again, pleased to be here with the other panelists to discuss where we're headed in the next century of treating type 1 diabetes.
Thank you so much, Felicia. And thank you also for bringing it down to the patient level with that example of the individual that you met who was successfully transplanted with islets from a donor. Well, you've heard from the three individuals who are actively working in developing therapies either for early or late stage type 1 diabetes. It's now my pleasure to introduce our final panelist, who really does not need an introduction at all. Dr. Francine Kaufman is a friend of mine and a tireless supporter of diaTribe, and someone whom I and the entire diabetes community applaud for her work as a pediatric care provider, as a passionate advocate for achieving equity for all individuals affected by diabetes, and as an effective researcher and administrator in the device industry, first at Medtronic and now at Senseonics, where she's the chief medical officer. She's a respected leader in the diabetes field. She's a professor emerita at the USC Keck School of Medicine and a former president of the American Diabetes Association. Among her many accomplishments was the winning a Telly and a Cine for a documentary on diabetes in which she starred. No Emmy this year, Francine, because it was in the past, but you might have been up for it were it available.
Well,
Fran, can you provide some perspective on the current status of diabetes as you see it, because you've been in this for a while, and what the future holds, particularly as we look to the promise of some of these new potential therapies?
well, I certainly can, and thanks, Alan. I really appreciate this opportunity. I want to thank Kelly Close for allowing me to be here today with these distinguished panelists. And I want to actually give a little perspective from my vantage point, being in this field for over 40 years, and the opportunity that I had early in my career to actually be in the early investigations on whether or not we could interdict the type 1 diabetes process. So in 1993, the type 1 diabetes prevention trial started, DPT-1, and it had really come right on the heels, if we can believe it or not, of the DCCT, the Diabetes Control and Complication Trial, that we had to do actually to prove that managing blood glucose mattered. And before that, and I'd been already in the field for a couple decades, we would actually argue about whether we really had to manage glucose or not, but we got the definitive proof. And then we flipped straight over into, could we really now fundamentally change that process altogether? And it was a wonderful trial. It did not succeed, although there were some signals about the use of both oral insulin or very, very small amounts of injected insulin in people, just as Henry had said, in these early phases of type 1 diabetes. Then DPT-1 morphed into TrialNet, and I had the opportunity to be a TrialNet investigator as well, where we started looking at more immune modulation rather than trying to get to the antigen, which we thought in part was insulin, that triggered this autoimmune response. We went through a variety of different agents, teplizumab, what Provention Bio is talking about was one of them, and many others have come to the front. And so you would think from 1993 till now, maybe, it is the right time for us to have finally found some things that might really have an effect for patients. It will only have an effect if patients are willing to enter clinical trials. We'll only know what might happen if their relatives or maybe even members of the general public are willing to be screened for the antibodies of diabetes. But I really do feel, and as the three panelists before me so aptly introduced, not only perhaps are we ready to interdict the process so you don't have full-blown diabetes, or if you do, perhaps it's modified for the course of your illness. And if we are already diagnosed, maybe it is the time to think of an intervention that can be almost like a cure. Which gets me to the story I wanted to tell. I was in my clinic at Children's Hospital Los Angeles a couple weeks ago with a new onset patient, a new onset 18-month-old child. And, the father kind of looked at the nurse educator and asked, well, this seems kind of primitive. Of course, we had a syringe in front of him and a vial of insulin. And where are all these advances? Well, there are tremendous numbers of advances we've made in the field. We've got, obviously, new insulins that have much better efficacy for patients. We have new technologies. We have perhaps the closed loop that actually can almost come close to getting as much of the time with glucose values in range that, certainly might mitigate any acute issues such as diabetic ketoacidosis or hypoglycemia or long-term complications from dysglycemia over years and years. So we've come a long way. We're now at this very exciting threshold. And, I would like to go back and find that father and tell him to, certainly watch today and to listen to, where we are at and maybe just a little bit more marching down the path, and we will have something that will be hopefully really revolutionized for people with type 1.
Thanks, Fran, for providing that perspective. I think those of us who have been in the diabetes field for as long as you and I have recognized some of the frustration of seeing these advances and not having them applied broadly and not as quickly as we would have hoped when we started on this adventure many years ago. But now is an interesting time. So you've heard introductory comments from each of our panelists. I think it's time now to explore some of the promise and some of the challenges, quite frankly, that we all face as we continue to apply contemporary science to delaying, preventing, reversing, or perhaps curing type 1 diabetes. We recognize that the goal is to improve the lives of all individuals at risk for developing type 1 or for those who already have a diagnosis, whether it's early in its course or after many years of treatment. And I think Fran well pointed out some of the challenges that having diabetes, we have resolved the problem almost universally of having patients not die from type 1 diabetes. Unfortunately, still happens from DKA, and it still happens from severe hypoglycemia. But that's a lot different than it was 100 years ago. But living with type 1 diabetes, as Henry said in his introductory comments, is not easy for anybody. It's not easy for the patient, particularly for children. It's not easy for the parents of those children. It's not easy for many of you who are listening to this this session on your computers right now. So we're going to explore some of the things that we can do together. And I think there is a bit of together in this that's very important. So some of the important differences between the time of the discovery of insulin and now are the challenges and expectations of clinical trials to determine the efficacy and safety of new treatments. Remember, when Banting and Best first isolated that extract from the pancreas, they didn't go to an institutional review board and start a randomized, double-blind, placebo-controlled clinical trial to inject Leonard Thompson, the first recipient of the Toronto insulin, with that injection. It was a very different era. And several of you have already mentioned some of the challenges of conducting clinical trials with these new therapies, not least of which is screening and identifying patients. But let's explore that in more detail. I'm going to direct questions to specific individuals, but this panel will be most exciting if others of you also provide insight. So don't hesitate to sort of jump into the discussion. Georgea, I'm going to throw the first one to you. And it's, how difficult is it to identify subjects for clinical trials, and what approaches might improve recruitment and the conduct of clinical trials? In your case, for early onset type 1 diabetes, but we're going to extend that to even earlier than that.
Yeah, absolutely, Alan. I think all of us touched on it a little bit. And I think as things have evolved over the last few years in type 1 diabetes research, I think the approach to identify patients has become more and more challenging, especially coming at the end of this pandemic, has really even introduced new hurdles that we really didn't have before. But one thing I think we all are committed to in the research space is evolving with those challenges and meeting physicians and patients where they are, and looking at this as more of an opportunity to be innovative, to really strive and devote ourselves to finding better ways to recruit patients, but also, expand our recruitment beyond kind of the isolated populations that we may just initially think about, to be able to target some of those underserved areas where we are not maybe able to recruit the right level of diversity in the specific disease area. So I think from some approaches that we've taken to improve recruitment and really focus on the inclusion of ensuring, diversity in underrepresented areas in pivotal studies, is really a deeper engagement with the physician community and the patient community to really understand what's happening and what's going on, and where we can meet these patients where they're at, how to make it easier for patients and their caregivers and their families. So I think one thing that we've really taken a step back and said is, let's talk to the groups that are really on the forefront of being included in these clinical trials as kind of our primary focus to really ensure that we're trying to identify the right patients and get them enrolled in these clinical trials. So really it's just starting at the physician and patient level, speaking with them one-on-one. And I think the other area is technology. I think technology has really advanced to help identify patients for clinical trials, but also help us where there are underrepresented populations from a diversity standpoint. And the technology platforms are continuing to evolve. There's a variety of third parties that are now coming into play. And I think one area for us as Dompé is really the engagement of new technology platforms. specifically focused on either the active referral patterns or the referral networks, where some of these patients, especially in the new onset population, but really across the disease continuum, can really help us in engaging those patients and also finding where these eligible patients are. And I think the other area, or kind of the last area that we've really focused on, is the real world data to really help us predict not only enrollment, but also the recruitment patterns, as well as really accurately depict how we can properly find these patients, recruit them into the clinical trial, and then assist them staying into the clinical trial, especially as we all know, some of these type 1 diabetes clinical trials are really long. And so not only is it identifying them challenging, but also keeping them enrolled in these clinical trials. So finding new technologies and advancements that can help these patients and their families stay in these clinical trials is also another big challenge that we really have focused on, of patient identification, but also the patient journey through the clinical trial, and using technology and platforms that really help with those two areas to help continue this research process that is so challenging, and now with the pandemic even more challenging. But as I think we all are in this group, super dedicated to just finding new ways to continue to make this work.
Great. Any of the others of you want to add anything to what Georgea said? I mean, I think one of the challenges we have, particularly with new onset type 1 diabetes, is the geographic distribution and the fact that so many of those individuals who have no idea they're at risk develop a disease state and go to their family physician or the emergency room in centers or hospitals that have no connection with clinical trials or academic centers. And they're essentially lost to the pool as we know it today. So how do we deal with that?
So if I can jump in, and building on what Georgea said, you know, the people living with the disease who should be most appropriate or benefit the most from clinical trials live in their home, and the clinical trials are happening in the clinic. And so until we can figure out a way, and I think George was alluding to this, that there are companies that are doing decentralized trials and doing them in the homes of patients, etc. But we have to recognize that unless screening is done on a systematic population base, we are going to be stuck in this situation over and over again. And, you know, I recently saw a patient with 2 kids. The dad has type 1. I diagnosed one of the kids, and then 3 months later, the second kid developed type 1 diabetes. And I asked mom, and I said, did you pick up on any of the signs and symptoms of your second child? And she didn't, or she did, and she sort of didn't want to acknowledge that that was the case because of how terrifying that must be. And so I think that even in the best of circumstances, as we look, and we can use big data and try to identify where people with type 1 diabetes live and where they get their care, the only problem with that is that really restricts us and limits us to a population that we know has a higher risk. That being said, we have to start someplace. And so I think raising awareness, speaking to the folks who are going to be the first to see those families, starting at the OBGYNs, the family practitioners, the diabetes clinical education specialists, and so on and so forth, the people who are actually speaking and engaging with people and know the families and are the sort of the main focus of where those families get care. You know, a while ago, I said to myself, how many times does a kid go to the pediatrician, is misdiagnosed, goes to the ER, is misdiagnosed, goes back home, is misdiagnosed? The pediatricians have urine dipsticks. You have a kid who comes in with a cold, there's a routine throat culture. Why not dip the urine? And so here's an example of where education may play a really important role in raising awareness that, you know what, this is something that's real and has to be looked out for. And it's as simple as doing a simple screening test, at least for newly diagnosed patients with a urine dipstick.
Yeah, I think, Henry, I think you've hit on some important issues there. I'd be curious if Fran wants to extend that a little bit with her extensive experience. But before going there, one of the personal, I'll give you my personal experience. I have a child with type 1 diabetes who developed disease at age 12, fortunately not younger. And I was in a denial phase when I went with him to a Boston Red Sox game and couldn't figure out why every half hour he was running, he needed to go to the men's room. And I mean, I was missing innings, for goodness sakes. It was really tough. But I mean, it did dawn on me as a diabetologist what was going on, and we were able to catch this before it got out of control. But the good news we have is that most, not all, but most children who develop type 1 diabetes within a family where there's a history of type 1 diabetes no longer develop severe ketoacidosis. It does happen, but that's much less common. The biggest problem, as you pointed out clearly, is that 90+% of individuals have no family history. And there we're missing people with those misdiagnoses, and they can get pretty darn ill before the appropriate diagnosis is made. Fran, do you want to extend that a little bit?
Yeah, I mean, I just want to add, I think screening, although we're all aware of screening for so many different diseases, is still hard. People don't necessarily want to know that they are at risk for something, particularly, and talking about the legacy of all of these other trials in type 1 diabetes, particularly when there wasn't something to offer other than a randomized clinical trial. So I think you could understand. And then, of course, going to the general public and screening for something that's still, a very low incident and prevalent disorder, particularly in children. And then figuring out when do we screen, who do we screen, how do we screen, who pays for the screening. It is going to have to be a public health endeavor. And, right now we're still in disease management kind of endeavors with type 1 diabetes. But, over the years, there have been, hundreds of thousands of enthusiastic individuals who wanted to be screened, who have entered clinical trials. I agree that we really need to look at diversity. One of the things about type 1, and if we say, why are we only on this VISTA now? This is a very heterogeneous disorder, right? It's not the same from necessarily one individual to the next as to what their genotype might be, what their phenotype might, what genes they have, how they present, what kind of symptoms they have, and particularly what kind of antibodies they may have. So, we do need much more experience. But I think when presented in the right way, particularly to families, they are willing to, for the most part, be screened. And then I think we have to really develop strategies for the general public on how to screen and who to screen and when to screen. But, we have that capability. Now we have to have the collective will. And I agree with, everybody. In addition, it's not the endocrinologist that we have to reach in that capacity. It's the generalist. And we've got to convince that it is a disease that we can interdict or at least put into a number of clinical trials that are going to be on the forefront to get to the right kind of answers that we need to get to.
Yeah, I think you're absolutely right. And there's an additional issue that you actually sort of touched on tangentially, because as new treatments, the first of the new treatments get approved that might delay the onset of type 1 diabetes in the preclinical stage, as they get approved, is that going to be an impediment for other clinical trials with different agents in terms of recruiting individuals into those trials? In other words, what's your experience as they weigh the potential of participating in a clinical trial versus saying, well, I really want to go on that already approved therapy that looks like it, has an impact?
I do think, and I've told you this before, Alan, I do think it's going to have an impact. You still have to be randomized. You still, will be using an agent that we don't know an answer for. Whereas if there is something, at least, if it's preventing a couple of years, if it's maybe, again, I think we have to look at not just delaying, but also what is the course of the disease after that? Is there less glycemic variability? Is there more time in range? So we need more information. And I would definitely plead with everyone in the field that these are not just 2-year studies or 3-year studies, and they're not studies that end when somebody gets diabetes, stage 3. These are studies that need to go on, because that's, to me, a much more important answer. If I get stage 3 diabetes, is that disease modified enough that it will be much easier to manage with the technologies we already have?
And not only does that take time, it takes a larger number of individuals to be able to assess it. And I think that's a real challenge. But now we've talked about early disease and some of the challenges there, some of the challenges of screening. Felicia, let's go to later stages of type 1 diabetes, because that brings its own set of challenges, specifically in the conduct of clinical trials. Now, you specifically have spoken about the first trials that Vertex is performing, which is on individuals who have, pretty severe disease with recurrent severe hypoglycemia or hypoglycemia unawareness. But how do the new technologies, such as the automated insulin delivery systems, the AIDs, increase the challenges of trial recruitment for beta cell replacement? In other words, what's going to win out here, technology or biology? And how much is the new technology going to impede our ability to test the new biology?
Well, I'm afraid I can't give you an unbiased answer to the question about biology or technology, because we're so excited about the idea that we can recapitulate that natural biology that we know works physiologically. But I think, to your point about clinical trials, I think some of the same concepts around clinical trials awareness, around clinical trials education, apply for when you're executing a study in people living with stage 3 type 1 diabetes. I think we need to make sure that the endocrinologists and other healthcare providers that these people across the country are seeing are aware of the clinical trial options that their patients may be candidates for. And that means making sure that people who aren't necessarily seen at large academic medical centers have the same kind of opportunity to participate in trials of novel therapies that they may gain great benefit from. And so I think that's a piece that we need to do a lot better at. Unfortunately, this is a disease of more than 1.5 million people in the US alone. And so there are many, many people who could benefit from a trial and could benefit from many different kinds of technologies that are being developed. And I'll come back to your comment about Banting and BEST. And, if I think about one of the reasons that it takes so long to develop these new medicines, it's a little bit of that double-edged sword. And one of the reasons it takes so long is that we, as drug developers, as scientists, spend so much time working preclinically to develop data, to develop packages of information about the particular therapeutic, working with our regulatory agencies to make sure that we have a product that has a potential benefit risk that makes sense for people living with the disease. And so that's a very different place to be than Leonard Thompson 100 years ago. And so we want to make sure that endocrinologists across the country and physicians across the country understand that about clinical trials, such that, again, we can make sure that access is equitably distributed across the country and globally.
Yeah, I think one of my take-home messages from this session for all of you out in the audience is clinical trials are really a good thing. It's how we advance science into the clinic. And you should talk it up, think about it, encourage others that you know to participate in clinical trials. We're not always talking about prevention, delay, or replacement of beta cells. All sorts of clinical trials in type 1 diabetes or type 2 diabetes can be very important. Okay, let's shift gears just a little bit. Georgea, you talked about the therapy that Dompé is developing in terms of altering the course of early onset type 1 diabetes. But from a clinical perspective, and you know this as a diabetes educator, and we all, those of us who are clinicians, know this from our own work, the diagnosis of type 1 diabetes, the initial diagnosis, can be pretty overwhelming, whether it's a child or an adult. I mean, it's just an onslaught of new information, and half of which, you know, is lost before it ever leaves the examination room. How challenging do you think it is to add yet another therapy other than insulin, you know, beyond insulin, into the early stages of treatment of diabetes, as will be the case with your development or with Henry's as well, in terms of what Provention Bio is seeking to do?
Yeah, and thank you for directing this question first to me, and I'm interested to hear what the panel has to say, because I spend a lot of time thinking about this. And I think, as many of us are aware, there are many studies of people with type 1 diabetes that really outline the emotional and interpersonal challenges that we really have to acknowledge before we can create new therapies in this space, and especially pharmacotherapy aspects of it, in addition to insulin and other therapies these patients will be on. So I think adding a new therapy in the earlier stages, specifically when a patient may test positive for 2 or more diabetes-related autoantibodies, at this point, we know that the immune system has started attacking insulin-making beta cells. The blood sugar levels remain normal, and there are no symptoms. It poses all these really interesting challenges. Overall, adding something new to insulin, which serves as kind of your base of care, needs to be thoughtful on a few aspects. And I think the 3 aspects that I continuously think about is the method of delivery and the dosing frequency, as well as really the difficulty of monitoring. And I think for us, specifically for Dompé, our commitment to kind of this challenging and overwhelming effects to these individuals, as we were taking our path, is first, can we develop a medication in an oral form to be taken at a minimal amount per day? And I think that's where latorixin, kind of being a new novel investigational medication, kind of came to fruition of being an oral therapy to really help minimize some of these challenges that these patients may face. And one other thing that we did is sticking to the BID, or twice a day dosing frequency, really has shown in data to be the highest level of adherence in this specific patient population and in many others. But our ultimate goal of this clinical trial, and whether latorixin treatment is effective in preserving beta cell function or delaying progression of type 1 diabetes in adolescent patients, is really to prevent the sequelae that drives the increase in insulin requirements, and hopefully be able to sustain patients within range to help minimize some of these challenges that these patients face overall.
Alan, if I can add something, you know, the issue, I think if we look at it across the spectrum of stages, so the first question on getting screening and what does that mean for the stress and people waiting for the shoe to drop, et cetera. Excuse me, it's a legitimate question. There's no doubt about that. But I also think that, you know, there's data to suggest that that level of anxiety and concern wanes over time. But, you know, we in the pharmaceutical industry, we make a lot of assumptions of how people will feel and how they'll react, and, you know, what's going to add stress and what's not going to add stress. And, you know, this reminds me of a time, and Fran, perhaps you'll appreciate this, that, you know, people used to insist that if someone's going to go on an insulin pump, that they need 6 months to be able to settle into the diagnosis and learn and go to educators and social workers, etc. And no one really asked the patient or the family, well, what's it like to go home with a vial of insulin that, you know, you're told too much can kill you and too little will make you suffer complications. And so challenging that paradigm and offering insulin pumps to patients at diagnosis was unheard of. Yet that was something that I thought should be offered to patients, since it's a better way of delivering insulin. So I think that's a really important point to make, that we should be careful with the assumptions because the data is all over the place. But if you look at screening by itself as a treatment to prevent DKA, and if we stop right there, I think this has been demonstrated in populations overseas. There have been a number of studies that have demonstrated a fraction of the patients who've been screened develop DKA as compared to those who haven't been screened for obviously a variety of reasons, including being followed more closely. So the overwhelmingness that we have when it's stage 3 and how we make those kinds of conclusions, I think we oftentimes do that on data that isn't that convincing. So I think if you say to somebody, here's a treatment that could potentially alter the course of the disease, and you say, here's a bottle of insulin, and you explain to them what the difference is, I think we underestimate the ability of patients and families to be able to use that information to make informed choices. And incidentally, when we talk about screening impacting on the conduct of clinical research trials, I think that raising awareness of screening results in everyone, including all the clinical research that's going on. So I believe that doing screening actually will encourage people, because not everybody's going to want treatment A or treatment B or treatment C for a variety of different reasons. So I know I sort of branched out in a bunch of different areas, but that's sort of how
You did, but you're going to get one more chance at a slightly different question, because I want to go a little semantic here, and then I want to also hear from Fran on this one specifically. Because, you know, we, in the 90s, when we talked about the decade of the cure, that was the JDRF big push for the 1990s, that they came, the 90s came and went, the 2000s came and went, the 2010s came and went. We're now, you know, approaching, I think, better science and better application. But we throw around words like prevention and delay. And I want to ask you how those differ from typical notions of a cure. And what does that really mean for people with type 1 diabetes? And also, to extend that, what does success look like for these therapies? And how do we know how long they might work? It gets back a little bit to what Fran said earlier about, you know, the duration of clinical trials. But Henry, I'm going to give you first crack at it. Then we'll go to Fran, and then I have a great question for Felicia.
I lost sleep over this question. I really did, because I started writing notes, and then I said, wait a minute. What does delay mean, and what does cure mean? And I said to myself, we can look at all kinds of different definitions. What a cure means to someone is an individual assessment of the impact of the intervention. So if one receives a whole pancreatic transplant and requires immunosuppressive therapy, is that a cure because they no longer require insulin, or is it an exchange for one disease for another? But I would really direct this question to people living with the disease, since these words mean different things to different people, and my responses would be both insensitive and do them no justice. So one thing I do know, and that is insulin is not a cure. That is, I think no one would agree that insulin is a cure. But I do think that, again, when we think about the nuances of cure, prevention, delay, you know, at the advisory committee that was looking at teplizumab, there were 181 people who spoke about what delay means to them. It was, if I had to actually read some of those letters, I don't know if I could actually continue with the rest of the interview. I mean, they were profound. So is it meaningful to them? Do they see that as being valuable? Is it the same thing as a cure? Do they see it as a week or 2 weeks of not having diabetes meaningful, etc.? So when I saw this question, I said, okay, I have an answer. And then I took a step back and said, it doesn't really matter what I have to say about it. It's really about what people who are living with the disease feel.
I think you're absolutely right. I mean, diabetes is such an individual disease. I mean, everybody lives it differently, and hence the potential of a particular treatment, monitoring, pump, prevention, delay, beta cell replacement means different things to different people. Fran, you've been at this for a while.
Yeah.
What do you think?
Well, I do want to say that I agree with Henry and you, Alan, but at some point we are going to have a definition of what is an endpoint that is either achieved or not. Otherwise, we'll be in a quagmire and we won't know really how to help patients. I think one of the things we also have to look at is the data from the Type 1 exchange and 22,000 people who, in 2010 to 2012, the mean A1C in this country in that group was 7.8. And then in, 2016 to 2018, it went up to 8.4. So all these advances, all these technologies, all these maybe, different insulins and everything else, it was a little bit before closed loop. But we're certainly not there with being able to, with what we have now, and with a burden on the patient, even getting close to normalizing glucose for most people. So I think one of the things that we have to look at, because we know that that's the issue, right? That's what that DCCT told us. That's what we know from the clinical experience of severe hypoglycemia, that we have to get closer to more and more people getting normalization of glucose. And, would that be a cure, Henry, if you had normal glucose, but you had to take immunosuppressants? Somebody's got to decide that. Would that be a cure if you have to have beta cell replacement every year, every other year, every whatever interval? Somebody else might need to decide that. But I think the minimum is that we've got to at least get to something that can come close to normalizing glucose and the other metabolic parameters associated with insulin deficiency or insulin, and then insulin administration that's not physiologic enough.
Right. And I think we could spend a lot of time talking about that. Unfortunately, we don't have that time tonight, maybe at another musing. But Felicia, I promise to come back to you because, all of us who have loved ones with type 1 diabetes, and I would suspect the majority of people listening to this session, have high hopes for a cure. And we've just discussed some of the challenges of defining a cure. But this is true for those with established disease, as are the hopes for prevention or cure for those at risk, but without the disease yet. How rapidly do you think the current advances in science address these hopes? I mean, how close are we getting to really making a difference? A difference meaning that, in your case, with what you're doing, that biology can take over from what technology is currently trying to do and doing a much better job of than it was even 5 years ago, but it's still a burden for the individual patient on a daily basis. So,
yes, it's absolutely the case, and your words, Fran, were so powerful about the burden and the limitations of what has been developed so far. And I think that's our job, is to continually develop better and better options and give patients that choice. I think what's really exciting for us is that the science really has turned a corner in the last few years in terms of our understanding of beta cell biology, stem cell biology, immunology, and those pieces we're now putting together into candidate therapies for the first time, going into clinical trials for the first time. And so I think that we are, of course, closer than we've ever been with this type of technology. There's for sure more work to be done, but when I look back even where we were as a scientific community 10 years ago versus what we understand, what we can actually do in a laboratory and a manufacturing facility today, it's really night and day. And so I certainly expect that the 18-month-old Fran you spoke about as he or she becomes an adolescent, that he or she will have an incredible increase in the types of therapeutic options available for hopefully transforming how they think about their disease.
You know, we get really excited about these advances, and we should, because they're extraordinary. On the other hand, I think most people listening don't recognize how many iterations one has to go through in terms of refinement of the approach. In the case of beta cell replacement, it's, you know, how mature are the cells? Mm-hmm. Are they encapsulated? Do you need systemic immunosuppression? Can you alter the cells so that you don't need immunosuppression? Are there other adjunctive therapies that can be used? How many iterations and how long do you think this is going to take? Not to discourage people, but let's put a dose of reality into beta cell replacement as a important. And we're not even talking about scaling of production in terms of, you know, the factories to produce all these little guys that go into transplantation.
Yes, absolutely. And, you know, of course, your point is so well taken when it comes to the timelines for drug development, the timelines for getting it right, both from the science perspective, from the clinical trial perspective. But I really do think we're talking about significant, meaningful advances in this field in years, not decades. And I think that's a different place to be than we have been in the past. And so I think that, you know, we will see, as you point out, multiple iterations of a cell therapy for type 1 diabetes that address these challenges to making it the right choice for a patient or the preferred choice for a patient, whether it's how long the therapy lasts, whether it's what type of other pharmacology, what type of immunosuppression or not you require, what type of surgical procedure is involved. I think that we'll be developing iterations on that make it better and better options for people as the years come.
Great. I want to thank all of you. You're not done yet, but I think it's time to turn to some of our participants who have floated questions for you. I don't think there's anybody here specifically, and I would like you to just jump in, and then we'll come back for one last question that I think is important, and we need to hear a couple of words from Kelly, because I understand we had some streaming problems at the beginning of this broadcast. So there's a question from Terry. Excuse me while I read this from my screen. I've had type 1 diabetes since 1972. Is there any hope that clinical trials might be opened up to us long-term patients?
An enthusiastic yes. And I would encourage you to speak with your endocrinologist or go to the JDRF clinical trials website, the diaTribe website. There are a number of trials ongoing that you might be eligible for. So appreciate the question and enthusiastic yes. Anyone
else?
Oh, I'm going to say an enthusiastic yes, too. Because I think as long as you have beta cells, and we know that people continue to make insulin and have beta cells into the 6th and 7th decade and beyond, there's hope for clinical trials, either by stabilizing, replacement, or other therapies that could potentially augment the ability of the beta cell to produce insulin and to preserve the beta cell function. I think that there is hope. Absolutely.
And I would add to that even in the area, apart from the topics we've discussed today, whether it comes to preventing complications or improving complications or just improving glycemic control with adjunctive therapies, there are many opportunities and many clinical trials being pursued. So for those of you in the audience, think about the issues that you have, and you can speak to your physician about clinical trials. And as already been said, check out the JDRF and diaTribe websites to try to find clinical trials that might be appropriate, or clinicaltrials.gov. All right, here's a really thorny one. I don't know that we're going to come up with a good answer for it, but it's certainly one that is very pertinent. And it comes from Michael. And the question is, are insurers going to be willing to cover the therapies? particularly along the type 1 diabetes onset treatment continuum?
That, if I could just start, that's obviously a big unknown. There's lots of things they don't cover. What seems to be necessary, and it's not just commercial insurance, it's our government insurance as well, which often even lags farther behind commercial insurers, but not always. This is where we're going to need some kind of definition of what the intervention is to do, and show that it's compelling from both a health aspect, ultimately also a cost benefit. And the fortunate part is diabetes costs a lot of money, and therefore reduction, particularly in the episodes of hypoglycemia. The type 1 exchange talked about 6% of people have severe hypoglycemia still, or DKA, 3% of the people have DKA. So there are a number of ways you could imagine that they will cover this over time. It's not going to be likely immediately. And I'm sure all of you, Georgea, Felicia, and Henry, will be working on that when you get an approved product. But that's part of the process.
And I think everybody should recognize that time is a key factor, not only in terms of the time to get coverage, but the time it takes to do these pharmacoeconomic analyses. In other words, how long do you have to follow an outcome to be certain that you have benefit? And one of the challenges of diabetes, type 1 and type 2, is they're lifelong diseases. Unless you can reverse them, which of course we're trying to do, the complications accrue, the costs accrue, lives are affected adversely, and it's a real challenge. There's a question from Forrest about what's stopping beta cell transplantation from becoming more mainstream. And I assume that means with the current technology, Felicia, and I know you can address that one.
Yeah, it's a fantastic question. And I would say there are two primary factors. There are a number, but two primary factors I'll highlight. The first is that this is a procedure today, before the advent of stem cell derived technologies, that used organ donation, so organ donated tissue. And those, as I think many know, are in very short supply, very scarce. And it's actually quite difficult to get a consistent, high quality product when you have an organ donor as the starting material for a therapeutic. And so that's a big one. It's also not yet an approved therapy, so it speaks to the reimbursement challenge that we were just talking about a moment ago. And then second, there is the challenge of the immunosuppression. For some people, those 6% who are living with recurrent severe hypoglycemia, those people, the trade-off may make sense. And for many who've undergone these islet transplants have made that calculus for themselves with their physicians. And so I think there is a sizable population of people living with this disease for whom it benefits, even with immunosuppression. But certainly to get to a therapeutic option where you're not having to make that trade-off will take resolving not just a high-quality cell source, but also an approach to protecting those cells from immune destruction without immunosuppression systemically.
Thank you. I think you're absolutely right. We didn't really have time to get into the whole question of immunosuppression, whether it can be more targeted or more general, lifelong or episodic. These are issues that are of prime importance to all of the therapies. Two of them are immunomodulatory. In the case of beta cell replacement, it's a matter of engineering the cells so they don't need immunosuppression. That's maybe going to be more difficult than we think, but certainly we have some great ideas with new technology. And one of the questions that we don't have time to get into because of specificity is, can you use techniques like CRISPR to redesign the surface of a cell? And that is being pursued, I can guarantee the questioner, with vigor by many, many different companies. I have a last question that I wanted to throw out, and then we're going to bring Kelly back on for a minute, and then I'll come on to close this out. But the question I had is one actually very similar to one that Charisse, our own Charisse Shockley, posted in the chat. And like every new innovation in diabetes care, there's a question of access. And given the major racial, ethnic, income, and geographical disparities in diabetes care and outcomes, what can we do to make sure treatments to prevent, delay, cure diabetes are accessible to everyone? And to be more specific, Charisse has asked, what's being done to build relationships with community leaders and clergy and community health workers to raise the awareness of some of these issues so that we can address them head on?
I'm happy to go first because I think this is one area that Dompé is extremely dedicated to. Being a new company in diabetes, I think we've really taken a step back, especially at this particular time, to look at where these underrepresented populations are and what the potential avenues would be to address those needs. And I think it really requires a much more demanding, sustained commitment and level of cooperation across the ecosystem to just be, as broad as I can without kind of saying specifics about what some of those tactics may look like, just the global ecosystem holistically in order to really be able to bring these clinical trials, but then also once they are approved to these populations that really need them more than anyone, but maybe don't have the right access to the type of care that either the clinical trials are in or where you could get access to these medications once they're approved. So I think for me and Dompé's commitment internally has been to really, one, get a better understanding of what our role is in this global ecosystem and what we could do as a company, and partner, to your point, specifically with patient organizations, community organizations, to be able to make a bigger impact. And we've started specifically with hiring some, specific organizations that have really great expertise in this area to really help us identify where we can start. I think because if we look at it from a holistic standpoint, where to start in the pharma company is always the hardest part, but we all know there's something that needs to be addressed here. And so we really took the approach internally to hire external counsel to give us an idea of where is the best starting point. And I think we're really coming up to some really innovative approaches to addressing this really larger health impact issue.
I agree with you. diaTribe has been, working with this over the last 5 years through the D series to try to think about how we can develop this grassroots effort to get diabetes a little higher priority on everybody's list, to make things happen with caregivers, with the general public, with policymakers, and it's really hard to make a dent there. Fran, you've again been at this for a long time, and you had experience also as when you were president of the ADA thinking about some of these issues. Any words of wisdom?
Well, just that what we knew before, I think, has come even into greater light with the COVID pandemic. And, it's just not acceptable. And there's multi-levels of strategies that need to be employed to look at social determinants of health, to understand the impact of the environment, race, ethnicity, access to medication, systemic racism inside the medical system itself. And I think it's starting, it's in its infancy. I think it will take quite some time, but I do think the leaders that I can see and listen to are committed to making change.
I hope you're right. It's time for change. And it's time for all of us, those of us on the panel and everybody out there in the listening audience, to get activated and think about these things so we can actually assure that new discoveries can be translated into better therapies and better outcomes for people. Kelly, let me turn it over to you for a minute.
Absolutely. Thank you so much, Alan. We just learned so much. The specificity of this discussion was such an honor to hear. Thank you for your expert moderating and insights. And so as we move to the community session and toward connecting to each other, I just want to express on behalf of diaTribe very directly the utmost appreciation to all of our generous sponsors who are supporting Musings today and who continue to support diaTribe's efforts to help people with diabetes, to help us be smarter and to help us live healthier and happier lives. And just a massive thank you in particular to our presenting sponsor, OneDrop, who's doing so much to engage all of us patients in our quest to be our best and healthiest selves as we move toward using data and technology as intelligently and optimally as possible. And that's on the way, right? It's on the way to prevention and delay and multiple T1D cures. Thank you to Abbott, our wonderful gold sponsor, Abbott Diabetes Care. The team really continues to move the world toward a more uniform use of CGM. And thank you so much to our silver sponsors, Lilly Diabetes, Xeris Pharmaceuticals, Zealand Pharma, AstraZeneca, Dexcom, Dompé Pharmaceuticals, Medtronic, Novo Nordisk, Provention Bio, Vertex Pharmaceuticals, and Sanofi. We wouldn't have been here today without your leadership, your sponsorship, and your support of diaTribe. You're remarkable organizations, and these are just companies, everybody who are just working so relentlessly and intelligently on T1D in particular. So thank you again for joining us, and thank you to our remarkable speakers, again to Dr. Alan Moses and the diaTribe team for bringing us Musings today. To everyone for joining us. And now I hope you'll click on the icons on the left-hand side of your screen to access the sponsored booths, the live one-on-one networking, and to reach diaTribe's and Sharice's community meeting to get her views and the views of fellow people with diabetes. Thank you again.
Great. Thank you. Thanks, Kelly. I also want to thank our four panelists. I mean, I had been looking forward to this discussion for some weeks now, and I was not disappointed. What an engaging group. Love to have further conversations with all of you. And I want to thank everybody in the audience for spending their time to learn with us today. As Kelly just said, Sharice Shockley has got the interactive community session starting just about now, so you can click on that icon on the left side of your screen. But I also want to remind you that there's another diaTribe event coming up next week. Next Tuesday, actually, July 20th at 7 p.m. Eastern Time, 4 p.m. Pacific. And that event is part of our D series, the D21 Lightning Talks, this time entitled Stigma and the Stories We Tell. We'll post the link. I think it just was posted to that event in the chat function now. And I want to thank you again. Thank our sponsors. Thanks, Kelly, for giving me this opportunity. Tom and the associates for pulling all of this together. And again, the panelists for a wonderful conversation. Have a great evening, everybody, and stay well.
Thanks, everyone.
Bye.